Welcome to the inaugural Data Blitz. We have 6 faculty members scheduled for today. We hope this Blitz series will aid you in preparing competitive proposals. This is going to be a weekly feature.
Krish, I'm sorry, Krish. I was not scheduled, but somehow I'm getting into it. Is it okay?
Absolutely. Everyone is welcome. The PVAMU research community is welcome.
Originally it was Dr. Samudu, but I just happened to enjoy it.
No problem. Everyone is invited. Don't feel bad.
It's so nice to participate and support.
Okay. Now, each presenter is allowed 8 minutes. I will give you a warning signal when you have a minute left. As you know, this is a blitz, so you are going to introduce yourself and present your research expertise and also give an overview of what you are doing. Everyone feel free to submit questions through the chat box, and the presenters will respond to you by email later. By the way, this session is being recorded, and if you have any objection, let me know, because we are going to record only the speakers, okay? And before I open the series with the first presenter, namely Dr. Youngblood, I would like to invite Dr. Rajan to say a few words. Dr. Rajan?
Yeah, thank you, Krish. I hope everyone can hear me well. Well, first of all, a very good afternoon to all of us gathered here. This is a new initiative from our office, and this is the inaugural research Data Blitz or colloquium. So to begin with, I hope you and your loved ones, they're all doing well and safe. That's the most important thing that's going around. To say a couple of words about this, this initiative led by our new Office of Research Advancement, led by Krish, is created in hopes to bring more visibility to your research and the research activities going on, and also to provide support and facilitate any growing research interest areas, particularly in interdisciplinary collaboration. That is another goal, is to connect faculty across campus among different disciplines. So those are the goals why this initiative is being created. And I hope that this will be an enjoyable session and also productive for all of you. I'm personally excited to learn about each one of your research so that we can assist you better with opportunities and networking within Prairie View and beyond Prairie View. So without taking any more time, I wish you all the best, and feel free to send us any feedback or questions that you may have. So with that, I'm going to send it, give the control back to you, Krish.
Thank you, Dr. Rajan. Now I invite Dr. Youngblood to give her talk. Let me get the slide. Dr. Youngblood, do you see your slide?
Yes, I do.
Please go ahead. Introduce yourself and your research interests, and say a few words about what you have been involved with, so that others can get some idea for their own projects later on.
Okay.
Please go ahead.
Good afternoon. Good afternoon. Hello, everyone. I'm Yolanda Youngblood, Assistant Professor of Biology here at Prairie View A&M University. My research interests include plant-based research, specifically Amaranthus palmeri, science education studies. I also serve as editor of Pursue, an undergraduate research journal, and I'm president of the newly reestablished Sigma Xi chapter here at Prairie View. Now, currently, I'm looking at Amaranthus palmeri. I'm looking at acetic acid as a growth control agent for Amaranthus palmeri. Acetic acid is a chemical agent found in household vinegar. Amaranthus palmeri is a dioecious weed that has caused significant crop loss to farmers in the southern U.S. Amaranthus typically invades crops and germinates before the crop, thus gaining a stronghold in the area. It outcompetes the crop. It comes up before the crop, thereby taking most of the nutrients in the area. It outcompetes the crop. Now, while Amaranthus palmeri has a high mortality rate in terms of its seeds, once that seed does germinate, it can produce up to 100,000 more seeds. It grows very fast. Now, previously, farmers treated their crops with traditional herbicides like Roundup. Now, these traditional herbicides had the chemical basis of glyphosate. Over the years, crops became resistant to glyphosate. So hence, traditional herbicides were not as effective because plants had resistance. They were no longer susceptible to the glyphosate-based herbicides. Amaranthus palmeri is one of those plants. So over the years, Amaranthus palmeri evolved two forms. One form is resistant to these traditional pesticides and one form is not. So the resistant form is glyphosate resistant and the susceptible form is glyphosate susceptible. Now, we've been looking at the growth habits of this plant in our lab. We know that 5% acetic acid does not work as a growth control agent on any weeds, especially not Amaranthus palmeri. We know that 20% acetic acid does work. 20% acetic acid is used commercially as weed farm, and there are some other brands of it. But we know that it's called agricultural vinegar, and it does work as an organic herbicide. But because we're concerned about buildup in the soil, we're concerned about lasting effects, the goal is to use less than 20%. Will less than 20% work? Our studies have found that 10% works as a growth control agent for Amaranthus palmeri, especially if you find it at a young stage. Now, we know that Amaranthus comes up before the crop. So if farmers spray these crops 2 to 4 days after germination of Amaranthus palmeri, if they spray that with the 10% acetic acid solution, it does cause death. And this death is within 72 hours. Most of it is gone within 24 hours. If you look at the graph, we see that the glyphosate resistant, in 24 hours, 100% of the seedlings were killed in a 24-hour time span. The glyphosate susceptible appears to be a little bit more hardy than the resistant form. 85% of it is killed in 24 hours. But again, all of them were killed within 72 hours. So 85% of the glyphosate susceptible is killed in 24 hours, and 100% of the glyphosate resistant is killed within 24 hours. We're looking at further studies. We're also looking at why that is. What's going on genetically that's causing this difference between the two forms? So those studies are ongoing, but that's where we are now. We're looking at why there's that difference. This is an organic herbicide that we're using. So we're looking at what's going on genetically right after treatment. What's happening with those cells at the genetic level. My other project is with Pursue. Pursue, again, is an undergraduate research journal. It's a scholarly journal whereby those Articles that are submitted are peer-reviewed by active scientists in the field. This gives students a platform to submit their work. Many undergraduates, they work with their professors for a year or so, and there just isn't enough time to put together their work and submit it to a journal. There's a lot of competition against other scientists, graduate students, and the like. This journal is strictly for undergraduates. They may submit their work. It's not just here at Prairie View, it's across the nation and beyond. But this is a platform for undergraduates to submit their work. So hence, this is what my lab is currently working on here at Prairie View. Thank you.
Thank you, Dr. Youngblood. Wonderful presentation. Thanks again.
You're welcome.
Our next speaker is Dr. Amarasekara. Dr. Amarasekara, are you there?
Yeah, I'm here.
Can
you hear me?
Your slide?
Yes.
Wonderful. Please.
All right.
Proceed.
Right. Good afternoon, everybody. I am Ananda Amarasekara. I am a professor in the chemistry department, and I will tell you a little bit about my research interest and the current projects that we are working on. I joined Prairie View A&M in 2003. So before that, I have worked in a number of other places, universities, as well as my training. I'm a synthetic organic chemist. I did my PhD in a long time ago. That was in the early 80s. So that time, my research area was synthetic organic chemistry, pharmaceutical synthesis. I did the Taxol synthesis, famous anti-cancer drug. So then have done many other areas as well as postdocs for several years. Then when I joined in 2003, I wrote many proposals sticking into my own field of synthetic organic chemistry. Maybe I wrote around 15, 20 proposals without any success at the beginning. And then the synthetic organic chemistry is very hard to do, need a lot of chemicals, a lot of resources. So then in around 2009, I changed the field, complete change in the field, tried to do renewable energy, biofuels type area. I moved, actually it started during 2009. There was a move in the federal government mandate of mixing 10% cellular ethanol to all the gasoline sold in the United States. So this time, there was at the beginning it was in certain states, but then it was expanded to everywhere in the United States to mix 10% ethanol to gasoline. So my initial work was to make cellulosic ethanol. So that's that time I wrote proposals to ACSPRF, USDA, and couple of proposals to like NSF. So just all that time, like almost after this change in the field, I got like each of these proposals funded. So so since that time, I'm lucky to get funding from the NSF since 2009, continuous funding for these renewable energy projects. And at the same time, I am working as a partner with or co-PI with Dr. Komalapati in the Center for Energy and Environmental Sustainability Bioenergy Research Group. I lead the bioenergy research group there as well. So the initial attempts were like developing a catalytic method to break down cellulose in biomass to make cellulosic ethanol. So there are current ethanol supply in the US is mostly coming from corn ethanol. So it's to mix 10% ethanol to gasoline supply, you need about 2.5 billion gallons per year ethanol. So about 98% of that comes from corn ethanol. Then there are some cellulosic ethanol plants as well, but still cellulosic ethanol is a developing field. There are issues in breaking the cellulosic chain into fermentable sugars, smaller sugars like glucose. So the developing catalytic method was the earlier priorities of our group. Then they are in around 2015, DOE changed their priorities to hydrocarbon fuels to see whether we can make a hydrocarbon fuel directly from biomass, like a drop-in biofuel instead of ethanol. So now the DOE and NSF priorities are to make hydrocarbon fuels out of biomass. So it is like something like our understanding of the fossilized carbon, like petroleum, hydrocarbons, coal, is they were like millions of years ago. It was the plant material fossilized under earth, and pressure and the temperature, it turned into hydrocarbon and coal type material. That's our understanding. So it is, if we can make a chemical method that we can do that thing in hours or days to make hydrocarbon material out of biomass. That is the basic concept of this type of research. So the biomass material, cellulose, hemicellulose, and lignin are the main component, and the cellulosic component or hemicellulose component, main like sugars, the basic formula is CH2O. Like there is crude formula is carbon to oxygen ratio is very similar. So, but the fossilized hydrocarbon or crude oil, there is no oxygen. It is basic formula is like a CH2. So you have to get rid of that oxygen to make it to a hydrocarbon crude oil. So it's not that easy as it sounds like. But so we were developing like catalytic methods of depolymerizing the cellulose fraction, make it to furine intermediates, and then building up again to like C10, 20 range crude oil range molecules, and then a hydrogenation step to make crude hydrocarbon that is compatible to the petroleum crude. That's the one aspect of this project. Other one is we know that about 50% of the crude goes to the polymer industry. So we can make polymers also out of that crude oil. That's the bio-based crude. So I think that's my basic research that shown in here. Okay.
Thank you, Dr. Ramasekaran.
Questions if you have.
Sorry, you finished exactly on time. Sorry, I interrupted and took away 10 seconds from you.
All right. Thank you.
Thank you. The next speaker is Gina, Dr. Chiarella. Dr. Chiarella, are you there?
Yes, I'm here. Yeah, yes.
Please proceed if you're ready.
Okay. So my name is Gina Chiarella. I'm a tenure track assistant professor of the Department of Chemistry. I did my PhD at Stony Brook University, that is also called State University of New York at Stony Brook. I graduated in 2006 in inorganic chemistry, is in the specialty of in organic chemistry with Dr. Stephen Koch. I did my postdoc at Texas A&M University in the area of inorganic and organometallic chemistry from 2006 to 2012. I worked with the Professor Albert Cotton. I was the last postdoc before he passed. And also I worked with Dr. Carlos Murillo. Finally, I worked with Dr. John P. Fackler. He retired. I closed the lab. I cleaned everything. And then I came to PREDIVIEW. I came to PREDIVIEW in the fall 2012. I got my tenure track in the fall 2018. What else can I say? Okay, well, I have 28 peer-reviewed journal publications, 36 oral presentations, I believe 36, well, 36 oral presentations around the country, maybe 2 or 3 outside of the country, but around the country is around 36. And currently I have 4 manuscripts in preparation. Hopefully I will have time in this COVID-19 to write them. So, okay, I will talk a little bit about my research, and so you can see in the screen, so a lot of letters and some few pictures. Okay, my research is focused in the synthesis of transition metal complexes with a special interest in the preparation of compounds with resemblance to biological structures and with potential catalytic activities. So then you see where I'm going to. My goal is to see a way to do remediation in the environment, to find catalysts that work in the way that enzymes do, to change the catalysts that currently we are using in industry, pharmaceutical industry, and also in general, for some ecological products that we can prepare in the lab and they can mimic the behavior of the enzymes. It is not easy, but nature is very wise and did before us all these catalysts. Transition metal complexes are compounds that they consist in metal atoms, or it can be also metal ions. Mostly are metal ions indeed. They are surrounded by molecules or even ions also that we usually call ligands. They are attached to the metal. This is why we call ligands, because they are attached to the metal forming chemical bonds. Those chemical bonds are usually coordination bonds. In these metal ligands, indeed, the interaction is between the metal that has empty orbitals and the ligand that are molecules or ions that they have long electron pairs, or you can have a pi electrons or pi systems, I mean resonance structures, and that electron density is donated to the metal, making this link in between the metal and those ligands. Now, in this case, what we can say that the metals are Lewis acids and the ligands are Lewis bases. These coordination compounds, they exhibit very unique properties, and those unique properties depends on the metal and the nature of the metal and the nature of the ligand. However, the predominant behavior of those complexes is due to the metal. What the ligands usually do, maybe I have some discrepancies with the biochemists, but this is what we in organic chemistry, bioinorganic chemistry, we believe, and I think this is a common understanding, that the ligands what do is indeed modify, tune the properties of the metal. So making this a very powerful tool if you want to prepare compounds for certain purpose that you have in your research. In my case, prepare compounds with catalytic activities. That is what I want. I started this project indeed 3 years ago, exploring diverse organic compounds as a possible ligands, and also investigating ligands that were already in use, but which properties as a potential catalyst or the luminescence properties they weren't explored at that time. So initially, well, when I started like 3 years ago, my research evolved into different lines, but not different, but 2 main lines. First, we have the synthesis and characterization of these metalloenzyme type compounds that could have potentially behavior as a catalyst. I was interested in the ones that were able to eliminate or indeed remove reactive oxygen species from the environment, like superoxides and peroxides, not only from the environment, but also from biological organisms. With this purpose, I did the synthesis with my students of 12 chip-based type compounds as a ligands and a couple more of non-chip-based compounds. We made the reaction with these ligands with the transition metal of the 4 row of the periodic table. So those transition metals are, in this case, the ions. We use ions. We don't use metals as a metal, but metal as an ion. Chromium 3, manganese 2, iron 3, cobalt 2, nickel 2, copper 1, copper 2, and zinc 2. We try first to optimize the synthesis of the ligands and then optimize the synthesis of the complexes and do the analytical characterization, determine the structure, of course, as far as we could, and also try the luminescence, electrochemical, and catalytic properties of those compounds. At the moment, we have done the spectroscopic analysis. I mean, the infrared spectra, UV visible, carbon, hydrogen, NMR has been done for most of these compounds. We have tried some structural characterizations. As you see in the picture, there is a crystal structure. This is a crystal structure of one of the compounds. We did this at Texas A&M in the same laboratory that I did my crystal structures as a postdoc. So we did this crystal structure. We have more samples to send there, to go with the students there and do the analysis there, but the COVID is stopping us. So this is in this project. We have a second sub-project, I will say, that is I'm doing this simultaneously with the first one. I'm working in the synthesis and characterization of metal complexes with amino acids. Especially, I'm working right now with histidine as a ligand. Histidine is a basic ligand, but a basic amino acid indeed, and is present in several enzymes. Is acting very well as a base, Lewis base. It binds very well to metals, transition metals. It is known that histidine works as a proton shuttle. What is that?
Dr. Giarella, you have a minute.
Okay, so the idea is we are trying to do the crystal structure of these compounds. We were unable so far because these reactions, the histidine is only soluble in water. But now we found the ability to have these complexes in methanol. We reflux the complexes in methanol, and then the compounds are prepared. So now we are currently working in the electrochemistry, testing the ability, trying to grow crystals for this. And I will be very happy if there is someone else that would like to collaborate or would like me to collaborate with them in the university. So.
There needs to be a global strategy.
Thank you, Dr.
Krish, this is me here. As the host, you have the capacity to mute everybody. I think there's feedback coming from somebody, I don't know. Looks like Dr. Parks. But you can mute everybody except the press center.
Oh, okay.
So I certainly do agree that a global strategy would be something very desirable.
Computers.
We've all come to enjoy living in a global community. Do you foresee freedom?
I'm still looking for the control.
Do it just a few weeks ago, or because of this pandemic?
So if you have the participant menu right there should be below the participant and unmute all button.
Oh, okay. Mute all. Thank you. Okay. No, okay. Can you hear me? I hope I haven't muted myself. Hello?
Yes, we can.
Yes, we can.
Yes, we can.
Okay, wonderful. The next presenter is going to be Dr. Kalubo Villa. I hope I have pronounced her name correctly.
Yes.
One second.
Okay, yeah.
Oh, sorry. Yes, please go ahead.
Okay, yes, that's good. And Good evening, everybody. And I'm Sumudu Kalubovila from the Department of Mathematics. I'm assistant professor in the math department. And my research area is based on image processing, computational mathematics, and mathematical modeling and medical image processing. I would like to give a brief information, detail about the image processing. What does it mean and how does it work? And image processing has a lot of areas. I think it's day-to-day life everybody is applying, but you are not using that word when you apply it. And just one, I think my research is based on, this paper is based on imaging, painting. I will give more details later regarding in painting, but I give another idea in image processing. One thing is image denoising. The denoising idea is, I think everybody did this one in your day-to-day life. When you take a picture, if you don't like to show your wrinkles in your face, I think some people, they use some application to smooth your face. That idea we call image denoising. That is also related to my research area. And the next one, edge detection, that is also another area in image processing. And the other area that is deblurring, I think that's, we face that one day to day when we are taking a picture. What is the deblurring image processing means? When you take a picture, sometimes your camera can be moved or sometimes the object can be moved. Then your output is, you can see some blur image. I'm in my research area, I try to remove that blur images. How do we remove that blur? That is also related to my research area. And the next thing is zoom in and zoom out. I think most of you use that one when you have image, how to zoom in and zoom out. But when you zoom in, sometimes you can see some squares. The problem is that method we use for zoom in, that is also we can use different mathematical model to getting a better image when you zoom in without getting squares. That is the idea of image processing. And this every image processing, I have to develop mathematical model. That is the reason I'm including one of my expertise area is mathematical modeling, because first I have to develop math model, then I have to check my model. When I'm checking my model, I'm using MATLAB because I'm writing program and for my model, and I'm checking is my model is good or bad. That is I'm using MATLAB. And another one is medical image processing. That is my the current and future that I'm working. I'm given more details in later what I'm doing, what can I do. And this is okay.
Hello.
And hello, can you hear me? Anybody question? No. Okay. And I published a paper 2019. Actually, I joined the peer review in 2018, and I should appreciate Dr. Hagigi for motivating me for publications, because when I joined 2018, he encouraged me to publishing papers. Thank you, Dr. Hagigi. And therefore, I published 2019. I published a paper that is modified finite difference and linear iterative PDE methods in digital imaging painting, and a Journal of International Journal, Multimedia and Image Processing. In this, my presentation in here, you can see that my paper, my work, and my results. And the idea of the imaging painting is when you have an image, and if you can see some part of the image is missing, then how do we fill that part? That is the basic idea of the imaging painting. And this is, we have a lot of applications. I know my presentation, my images are too small. And if you can maximize or zoom in, yes, once again. And you can see it's larger. And the fun thing is you can remove some object if you don't like look like just an example. When you are taking a picture with your friends, in future, if you want to remove one of your friends' image in your picture, I can apply my model to remove that one. That is, we call object removing. And the next one is we call photo restoration. That means when you have an old photo, or if you have a piece of the photo, not the single photo, you have piece. And I can, using my method, I can build up the original photo. And the next thing is text removal. And I think that is in my presentation. I think everybody is left-hand side under the introduction. And normally, sometimes you can see some text in front of the image, but if you really like the base background image, you can apply in painting technique to remove the text. That is the application. And under the method category, I explain what is the method I use and what is the existing method. And the left-hand side arrow shows the existing method, and the right-hand side you can see one arrow, and it is divided by two parts because that is my method. I try two different techniques and two different models to develop and to in painting, and that is the just rough idea about my method. And under 2.1, I am explaining the existing, what is the method people now use. I think everybody know you have Photoshop, and you guys think, why? What is the difference? That is the idea of the Photoshop. What is the idea they use in Photoshop? The math behind the in painting. And in my method, in here, we use partial differential equation. I think people, you guys know, heat diffusion equation I used here. Everybody know heat is transferred from higher concentration to lower concentration. I think I mentioned I have the outside information. One of the area information is missing. I am using this outside information to fill this gap. That is the higher concentration to lower concentration. I am moving my information using my mathematical model. That is the basic idea. And here, that under 2.2, I explain what is my method. I'm explaining what is the existing method. Existing method, the people, they use entire outside data and they fill entire gap in one time. But my method, I am filling layer by layer. I am using entire outside information and fill one of the layer. I hope so everybody familiar with the images. Images means it has a pixel. And I think you guys can see when you save the images, it says 256 by something. That's size of the image. Each image, it has pixel. Each pixel has some value. The combination of these pixels that you can see image. If you can zoom in your image, you can see small squares. That is the pixel. Sometimes if you have maybe old computer, sometimes you can see that when you zoom in your image, you can see squares. Those squares we call pixel. And here in my method, I am not filling entire thing in using one time. I am filling by layer by layer. And also, when I am filling this layer by layer, I am use my new method. That is, I'm calling that is the 5 points, the modified 5 points stencil method. That is, I call my method. And also, I am using the linear iterative method.
Dr. Kalabovila, you've got a minute.
Yes, sure. And I'm using linear iterative method to fill in this, applying for imaging painting. And my results, it's showing. I know it's very tiny and it's showing. And what is my output with my application? I think you guys can see it is successful. And some images you can see line. I use the different diffusion inductivity. And the one thing I want to mention before I finish, I'm using, one of my manuscript is ready to submit. Maybe the next month I can submit it I can submit my manuscript that is based on error analysis. And the next one I'm using medical image processing. I really like if anyone in the peer review, if they would like to work with me in medical image, that I have idea to medical image processing. If anybody like, I invite them. Yes.
Thank you, Dr. Kalabovila. Very interesting presentation. Thank you.
You're welcome.
Our next presenter is Dr. Pulikartara. Dr. Pulikartara, are you there?
Yes. Hello.
Oh, please proceed.
Hello, everyone. My name is Dr. Pulikartara, and I am an alumni from Prairie View. I got my degrees in physics, chemical engineering, my master's in engineering. I did my PhD at Rice University in applied physics in the Department of Chemistry. And after that, I had done a postdoc at University of Texas working with cancer research. Then I went off to the oil field. So definitely, I've been seeing some presentations. I'd love to collaborate. I've worked with nanoparticles as well as microwave technology for the upgrading of heavy oil in Canada. Then I came down and worked with some more nanoparticles in other aspects of creating some different type of chemistry for downhole research. After 5 years in the oil field, then I went to Baylor College of Medicine for a couple of years, again, dealing with nanoparticles and this time pancreatic cancer research. Was heavily involved in microscopy and different type of cancer cells with different types of modified nanoparticles. After that, I've been at Prairie View as a lecturer in the physics department for about 3 years. And I have current ongoing collaborations with Dr. Carson at CARC and Dr. Yuri Makiev at Mayo Clinic. And together we're starting to do some continuation of the project started at Baylor to see if we can come up with a pre-treatment for chemotherapy of gemcitabine. Initial results from Baylor seem to indicate that something was possible, so I'd like to understand the mechanism. We believe it's some interaction going on with the serenol group on top of the fullerene C60 structure. And so what you see here is the figure on the right where it says C60-SER is the functionalized fullerene or little nanoparticle. It's 1 nanometer across, this little soccer ball that you see. And we have an idea that it is interacting with the DNA amino base pairs. We're not sure of the exact mechanism, and that's the part that I'm very much interested in. You see a little figure in the middle with the word gemcitabine. That's a common chemical structure of chemotherapeutic medicine. It's used to treat pancreatic cancer as well as other types. And so that is the basic idea of our first project. The second project is with Agriculture Department, Dr. Amapin, and collaborating with Dr. Osborne Lee in chemical engineering to see if different modes of delivery of nitrogen would make an effect on plant growth. We have our little experimental plan here on the right-hand side in the golden graph. The middle graph that you see was just the effect of dosing of gemcitabine on the different types of cancer cell lines. And this is the project, part of the project that we hope to continue in Dr. Carson's lab. And another one that I'm interested in doing is involving atomic force microscopy. And this is more about examining the role of van der Waals forces between different nanomaterials that have a different aspect ratio. For example, this little soccer ball that you see on the right-hand side has an aspect ratio of length to diameter of 1:1. So its ability to be drawn towards each other is also dependent on this aspect ratio. Nanotubes, for example, have an aspect ratio of 1,000, meaning that the nanotube itself can be up to a micron over a diameter of only 1 nanometer. So these are the 3 projects that I'm involved with. And I've published on different types of nanoparticle projects, including sensors, polymer nanocomposites, some oil field applications, and I chemically functionalize different types of nanoparticles for various applications. And I'm very much open to collaboration. Another thing I had done at Prairie View was the formation of NanoX. It's a group of scientists, engineers who have interests in nanotechnology. And I've had a variety of professors come from various fields. It's an interdisciplinary group in potentially coming together and collaborating on different types of projects together. Some we have started and hoping to continue this NanoX effort online starting in the summer. But it's been successful so far, and we want to have it continue. And so for these main 3 projects is what I'm doing now. I'm also involved in science education, and I'm like a unofficial co-advisor of our little physics group. And I'm looking forward to collaborating. As I said, I have experience in aerospace. My nano experience started at Prairie View as an undergraduate. And so for years, I was working under NASA at Prairie View, then to oil field, then to cancer research. And I'm looking for collaborators to join me in these efforts to expand nanotechnology collaborations at Prairie View.
Thank you, Dr. Pulikatara, for an excellent presentation. Appreciate it. Thanks. The next speaker is Dr. Allen. Dr. Allen, are you there? Dr. Allen?
Yes, I can hear you, Krishnamurthy.
Oh, please proceed.
Yeah, sure. Thank you very much. Hi, this is Dr. Jobar Alam from Civil Engineering Department. And I have joined Prairie View in the last fall 2019. I'm a very new faculty here. And I have my specialization in geotechnical engineering, especially in slope stabilization and infrastructure management using recyclable materials. And also I am greatly involved in sustainable waste management and landfilling. So today I'm going to present a few of my research that I have done for the first few years, and also some of the proposals I am working on. So if we look at the first picture at the top, we can see that the waste have been dumped in the landfills, and those are exposed to the environment. So before 1980s, before the application of engineered landfilling system, Environmental Protection Agency and many other state regulators, they used to use the traditional landfilling system or the traditional cover system. So once your landfill reaches the limit height, then you have to close the landfill with the cover. And that cover is basically with soil. And that soil will have to be, sandwiched between several layers of geosynthetic materials and other layers. And these things were working properly, but with the time, the performance of these type of things actually, degraded. So what happened, EPA and many other state regulators and researchers, they started, adopting some new types of cover system. And one of the most promising method is water balance cover system or evapotranspiration cover system. And this is basically a natural process, and that works with the natural process, which is when it rains, the rainwater is basically accepted by the cover soil, and that rainwater is converted to 4 components. That is a surface runoff, one is soil water storage, that is whatever water that infiltrates through that cover that is stored in the soil, and some part of the water actually percolates through the soil. And that water, if it mixes with the waste, what happens, that actually accumulate at the bottom, and if the bottom of the landfill is not properly constructed, eventually it will mix with the groundwater and it will be life-threatening for the society. So it's a very important process. At the same time, the main problem associated with this is monitoring. So many landfill operators and EPA, they recommended to use this, but they don't know how to monitor that if this kind of cover system is working properly. So, and some of the researchers, they recommended to use sensor system. Sensors is very good, there is absolutely no doubt, but the problem is they actually, work for maybe 3-4 months or maybe at max 6 months, because the environmental condition, they don't allow the sensors to live for a long time. So what I suggested, that if we use geophysical method, that's a very simple method, and we can have information in the landfills, and landfills is huge in the landfills throughout a long distance. So resistivity imaging is one of the geophysical methods which is very promising, and the methods we are recommending for the landfill operators, they will have two-way advantages. One is they will be able to know how much water or how much precipitation is being distributed in the cover soil, and also at the same time they will be able to know that one of the main component of the water balance cover is plant, soil plus plant. So how the plant system or the roots of the plant are working. So they will be able to know both. So if we look at the profile, that colorful contour profile, we can see that we have red colors and blue colors. So that red color is basically high resistivity. That high resistivity means actually there is very less moisture or very dry soil. And where you can see there is blue and green zone, that is basically the soil with moisture. So it is giving us the sense that the upper soil layer is getting dry. So if there is next precipitation event, that soil will be able to hold that water. And also, it also gives us the sense that how deep the root system of the plant is. If we look at this, the evapotranspiration process is a component of two things. One is evaporation, that is surface evaporation, and another is transpiration. And if the plant root system works properly, the transpiration component is significant. So if we look at this, that upper 1 feet to 1.5 feet of the soil is red, that is very dry. That means the top 1 to 1.5 feet soil is occupied with plant root, and that's why the evaporation and the transpiration process is more effective there. So we recommended this method, and many of the landfills have already adopted using this resistivity imaging method, and they are monitoring their landfills very well. And wherever they see that at the bottom of the cover there is many water, they take immediate actions. So that rainwater or precipitation, they don't infiltrate through the soil and mix with the waste, which will eventually mix with the groundwater. So that's one of my recent research we are adapting, and also we are in the process of writing few proposals. And also on waste management and landfill cover system, I'm in the process of writing a book. Hopefully it will be published within 1 year. My other part of research is sustainable geotechnical infrastructure management. As we know that the landfills, they accept a lot of plastics. So the plastics is basically non-degradable, so they pose a threat to the landfills. They occupy a lot of volume. At the same time, they don't degrade. So if we take this plastic out from the landfills and make some kind of pile system, which will we use in the slope system, that slope will be stabilized using the plastic. So it can give us two-way advantage. One, we are taking the plastics out of the landfills, and second, we are stabilizing the geotechnical infrastructure. So this two-way advantage, already I have one proposal in the underway, and a few more is coming. And another, my interest of research is pavement engineering and pavement distress and engineering. So as we know that in Dallas, Fort Worth and Houston area, we have very worst clay in terms of geotechnical term, that is worst clay. That clay property is when it accepts water, it automatically swells. And when it dries during the summertime, it shrinks. So this swelling and shrinking effect in the soil, it causes the pavement to shrink. And what happens, there is a crack in the pavement system. So what we are recommending and planning to implement is we will apply some geosynthetic material at the edge of the pavement. That geosynthetic material will route the precipitated water to the ditch so that it cannot mix with the soil underneath the pavement. So in that way, the pavement will have a long-term sustainability, and it can have a very riding smooth, and there will be no crack in the pavement. So yes.
1 minute to go, please.
Okay, so two of the proposal is underway, and two of my publications is also underway on the pavement engineering, on recommending for geosynthetic material for using in pavement materials and pavement engineering. And also one of my research interests is image analysis in geoenvironmental engineering. I believe that Dr. Sumudu, she already talked a lot about image analysis. So definitely it will be a great to talk to her and do something else regarding image analysis. But my perspective is different. She does, I believe, very basic things. By my part is application. I don't do very basic courses in image analysis, but I apply this method, image analysis, for the geoenvironmental engineering system. Thank you very much.
Thank you, Dr. Allen, and thank you all. Thanks to all the presenters and all the attendees. I hope the inaugural Blitz was useful to many of you in terms of finding opportunities for collaboration. I appreciate the response. I hope to see more of you next week at the same time. Thank you.
Thank you.